Good morning. Okay, let's continue on in chapter 5. And so we're at about 5.11. And so in 5.11, we're kind of doing a switch here because up until now, we've been looking at meiosis and understanding the ploidy level of those cells and the process of it. So the question becomes, well, can it ever go wrong? And it does. So here would be a classic kind of example that could come to a geneticist's office. Jason and Laura are pregnant with their third child. They're both over 35, so they're opting to have an amniocentesis. The doctor comes back to them with a karyotype, and that karyotype shows 47 chromosomes. If you were their counselor, how would you explain these results? Okay, so up until now, we're looking at our somatic cells and our body cells, and they've got 46 chromosomes, 46 total, 23 pairs of chromosomes. So what can go wrong? Well, when we start talking about genetic abnormalities, there's kind of two levels of it. There's a level that occurs at a chromosomal level, and that's what we're going to address right now, is on a chromosomal level. So that would mean really big changes to the number of the chromosomes, changes to the size of the chromosome. And then there's also genetic abnormalities that occur at a gene level. And so that would just be a mutation. When we think of genetic mutations, that's just a change to one recipe that's on a chromosome. That's really different than adding an additional chromosome or losing an additional chromosome. So when we start looking at those that occur at a chromosomal level, there's not a whole lot of them to study, but what they would look like would be too many chromosomes. Where an individual, instead of having 46 chromosomes, they may have 47. So they have too many. Or you can have too few chromosomes. So instead of having 46, you might be missing one and only have 45. Another thing that can happen to the chromosomes is that they can break. And so if you think about the process of crossing over and you think, yes, the DNA breaks and it switches position with its homologous pair, imagine if it breaks and the piece, its intention is to switch over during crossing over, but instead it gets lost. And so now that chromosome is shorter. So it's missing a ton of information. Or another thing that happens is when crossing over is taking place, the chromosomes break, but they adhere onto completely different chromosomes. So that's another major problem. Because if you think of our chromosomes as being cookbooks, and we talk about them as being the cookbook of life, what we're saying here is in one case, I have an extra cookbook. And your cells are going to read these cookbooks. And so if I've got too many of them, I'm going to be making too many of the products. Okay, too few chromosomes is, imagine now that you're, using these two cookbooks, but you're lacking one altogether. And so now you're missing half of your recipes. So the concentration of the product inside the system can be too low. You need both cookbooks, both chefs to keep making out what those recipes are coding for, and you only have one cookbook with one chef. And so you might be lacking the appropriate level of the amount of proteins or product that chromosome is coding for. And then chromosomes do break, and I've put in some karyotypes of some broken chromosomes so that way you can see what that would look like. And when they break, if they don't join on to where they're supposed to join on to, they just get lost over time. So it's like taking your cookbook, ripping out a whole bunch of the pages and throwing them away. Sort of has the same effect as having too few chromosomes. You have the chromosome there, but it's missing a whole bunch of the recipes. And then, of course, if when they go to cross over, they go to the wrong location, this is called translocation. if it goes to the wrong chromosomes and adheres onto that, again, it's like taking your recipes, like maybe you have a cookbook that's the recipes for all of your baked goods, you know, all your breads and your pastries, and you grab a whole bunch of those recipes and you shove it into your barbecue cookbook. You would never even know to go look at this other chromosome. So having your genetic information in its correct location is really key. So the incorrect crossing over or translocation is very, very significant. Okay, now all of these are rare. There's not a whole lot of cases where, you know, it's just one after another, after another, after another. And the reason why is because we're talking about huge amounts of information. It's not the same as a mutation. So when we talk about a genetic mutation and a change to one recipe, I might be lactose intolerant because I'm missing that gene, or that one gene has mutated, but that doesn't change a whole bunch of features about me. I've just got one bad recipe. You might not even know that to be the case. So when you talk about conditions like these that I've just described above, then they're really rare, because for the most part, if some of these chromosomes, you get too many of some of the chromosomes, the pregnancy won't even take. And so that could result in a spontaneous miscarriage. And so a lot of these are really life-threatening. And so because of that, they get filtered out, and there's very few of them. Okay, and then the other thing that's true about all of these is that they are phenotypically noticeable. So remember what phenotype means. That means my physical appearance. And sometimes those physical appearances could be internal, like you don't actually see them, but you might say, oh, well, the liver is smaller than normal. That's a phenotype. My liver is the physical manifestation of my genetics. And so if it's smaller than normal, that is phenotypically noticeable. Okay, and so all of these conditions are all phenotypically noticeable. You're not going to not know when one of these occurred. Okay, so remember our karyotype. So we've been looking at these a couple times, and remember what they're good for. They allow for us to see the chromosomes. And what we're talking about is changes to the chromosomes. And so what's nice about a karyotype is all of these would be visible to us in a karyotype, because the chromosomes are no longer going to look like pairs. Things are going to be missing. So again, here's a normal karyotype. You have 23 pairs of chromosomes. And so that's what technicians do. Okay, so how is it then that somebody can receive too many chromosomes or too few? All right, so non-disjunction is a process that can account for those two situations, where it's too many chromosomes or too few. The process is called non-disjunction. And so trisomy 21 is a condition where an individual has 3 chromosome number 21s. The resulting condition is called Down syndrome. So non-disjunction that produces that occurs during meiosis. And so it occurs during the production of the gametes. And during the meiosis, you end up having too many or too few chromosomes inside your gametes. The way that that occurs is either by the homologous chromosomes failing to separate during meiosis, or sister chromatids fail to separate during meiosis. So if we look at the normal, here's our review of meiosis. Remember, our chromosomes come as a homologous pair. Here's my cell that's going to undergo meiosis to produce 4 gamete cells. And keep in mind, this is the normal situation. Okay, and so your chromosomes come in pair. The DNA replicates themselves. And in the process of meiosis, you are separating those pairs of homologous chromosomes. so that chromosome number 1 from dad ends up in a separate cell than number 1 from mom. Then in the second round of meiosis, the sister chromatids are being separated from each other, so that a sister chromatid of dad's number 1 ends up in one cell, and dad's number 1, its sister chromatid, ends up in the other. This reduces the amount of genetic information, and the way that it's reduced it is that each cell has one member of the homologous chromosomes, and this is completely normal, the way that it should look. So those chromosomes, in order for it to be normal, notice that our homologous chromosomes line up side by side in the middle of the cell. During anaphase 1, the homologous chromosomes get pulled apart. Then the cell splits down the middle, and you end up with 2 cells each with one member of the homologous pair. Then in the second round of meiosis, then the chromosomes line up down the middle of the cell, and sister chromatids are pulled apart. And this is the normal way in which it's supposed to occur. So this is just review, because we're in 5.11 now, and this is 5.8. So this is the normal. So what happens then that, things can go wrong? So non-disjunction is when these chromosomes fail to separate properly. And as a result of that, if you track your gametes will either have too many or too few chromosomes inside of them. And it looks something like this. See, non-disjunction can occur in the first round of meiosis. So in the first round of meiosis, homologous chromosomes are supposed to separate. Oops, sorry, I'm doing the second round. So in the second round of meiosis, so our homologous chromosomes have separated. They've ended up in separate cells, but in the second round of meiosis, notice that here the sister chromatids failed to separate, and both of those chromatids ended up in one cell. That leaves this cell without a chromosome number 1, or whatever chromosome we're talking about. Notice that when it occurs in the second round, these 2 cells can be completely normal. Just because this one experienced non-disjunction has no bearing on the fact whether or not this one will also experience non-disjunction. So it's just as likely that this cell is going to go along just perfectly. And when that cell then gets fertilized by a sperm cell, it's back to its normal ploidy level. And this one would be back to its normal ploidy level. So if this were your gamete that you produced and you got pregnant this month, your baby's going to be completely normal. And you're not even going to know that non-disjunction occurred. However, if this cell is the one that becomes fertilized this month, then what will happen is your baby will have too many chromosomes, and that's a condition called trisomy, because tri means 3. So you end up with 3 chromosomes, whatever chromosome number it is. If this cell gets fertilized, and it can, if that cell becomes fertilized, it has the chromosome, whatever one we're tracking here, it has it from dad, but it's missing the one from mom. Because it's missing the one from mom, this is called monosomic. So mono meaning 1. And again, if either of these are, the ones that get fertilized and produce an offspring, you are going to notice the effects. Okay, now look at non-disjunction that occurs in the first round of meiosis. And in the first round, homologous pairs fail to separate. And so both homologous pairs end up in one cell. This cell is completely lacking. When the second round goes, sister chromatids separate, but you still have too many chromosomes inside that cell. These cells are lacking a chromosome. And if you compare, your cells here, you'll see that all 4 of them are affected. Notice here, only 50% are affected. And in this case, when non-disjunction,
happens in the first round of meiosis, where homologous chromosomes fail to separate all 4 of the GAN